Hey guys, so today we're going to talk about Roche 4-5 sequencing. So when it comes to the name of this method, there's nothing special to it. It just comes from the name of the company, 454 Life Science Company, and the author Roche put his name on it. And the importance of his method is that this technique was able to sequence DNA in parallel. And what that means is just that you can run millions of copies in a day versus like Sanger sequencing where you did individual sequence reactions. Roche's technique was one of the first of the next generation sequencing methods. It was involved with the Human Genome Project as well, and it also helped discover the cause of honeybee disappearance as well as new approaches to fight infectious diseases. It's important to note that this method was introduced in the early 2000s, so keep that in mind when we talk about the quote-unquote new techniques that they used in 454 sequencing. With the development of 454 technique, it was very thought out and thought to be innovative. Authors use pyrosequencing, which we should recognize from our lecture, but it is a method that's based on the detection of light that's emitted when a nucleotide is incorporated to a sequence. Now, they did face an issue of detection, but by adapting commercial-grade astrological cameras, they were able to view images in real time. Those cameras are called CCD cameras. So another challenge that the company faced with them making billions and millions of copies was somewhere to load them. So the 454 company manufactured a plate with millions of wells by just etching thin slides from a fiber octave block. So we use those wells a lot, so we can thank this company for that. So next we can get into the meat of this, how the process is carried out. Firstly, a DNA fragment is chosen and then it's cut into shorter random fragments by physical shearing. So then after you get your cut up randomly sized fragments, you would add adapters to them. There's two adapters in this process, usually called adapter A and adapter B. Once the adapters are added to the DNA, you can now put them on your capture bead. It makes a complementary strand. And what that means is if on your regular strand, the black one, if you had a A, you would make a complementary strand of a T. And if you had a C on the black strand, you would make a complementary band of a G. The next step to achieve those repeated strands on that bead would be emulsion PCR. So in the next clip, there's a very helpful video that explains emulsion PCR. The clip starts after a process of denaturation, which is done in emulsion PCR to take those double-stranded DNA and turn them into single-stranded DNA.
These beads are then mixed in emulsion oil in order to form fine dispersions of minute droplets and separate each of the fragment bead complexes. This allows for the amplification of each sequence simultaneously without contamination. Microreactors are fragment bead complexes in the PCR reagents in the emulsion miscals, or droplets. Each of these microreactors are ready to start amplifying the sequence. Primers are used as a starting point for DNA synthesis. DNA polymerase, the enzyme used in the body to copy DNA, is then used to add the nucleotides, or the dNTPs, from these primers. Buffers are added so that the pH is at optimum level for enzymatic activity. Miscals produce 1 million copies of each DNA fragment on the surface of each bead.
So after emulsion PCR, the amplified DNA attached to the bead is then added into one well. And those wells are on a plate called a pico-titer plate. So after adding your amplified DNA, you would then add polymerase, ATP sulfurase, luciferase, and a P-rase. And this is all added for the purpose of detection when it goes into the machine. So we can see here that we do have our strand, and this is just going to be a demonstration of what happens inside the machine with all those additives that we just did. So that there is going to be DNA polymerase. DNA polymerase adds a new strand. Let's say, for instance, that new strand is a T. So once DNA polymerase adds a new nucleotide, it releases a pyrophosphate. Once the pyrophosphate is available, ATP sulfurase engulfs it and converts it to ATP. Once the ATP is released, it joins with luciferase. And once the two are joined together, it gives off light emission. And that is what the CCD camera picks up and converts into a graph that we can analyze our data with. The graph looks somewhat similar to this illustration, but here is an actual graph. So the graph is to be interpreted by looking at the y-axis and correlating it to their colors that have been shown. So for this example, we can see that there was a very large amount of G's if you look at where it goes up to the 4, versus where some of them are very, very low counts of, let's say, T's or A's. And as well, sequences of DNA can be read from this graph. As you see on the right-hand corner, they do have a sequence put together just by correlating it with the colors. So it's very beneficial.